Welcome back to the Deep Dive. Today, we're tackling one of medicine's most profound challenges. How do we get powerful therapeutic genes directly into the very cells that need them? Imagine having a disease caused by a missing or faulty gene, and the solution isn't a pill or a surgery, but a precisely delivered genetic instruction that can fix the problem from the inside out. This isn't just science fiction. It's the ingenious world of gene therapy, built on the shoulders of molecular vehicles that are nothing short of incredible.
That's absolutely right. At its heart, gene therapy is really about correcting genetic defects or maybe introducing new functions into cells. But to do that, you definitely need a way to transport the therapeutic DNA. These transporters are what we call vectors, sort of the molecular delivery trucks. What's truly fascinating, I think, is the sheer diversity of approaches scientists have developed. From repurposing ancient viruses to designing completely synthetic carriers from scratch.
And that's our mission today. We're going to embark on a deep dive into these different gene delivery systems. We'll explore the highly engineered viral vectors, move to the more controlled non-viral methods, and even venture into the cutting-edge jumping gene technologies known as transposons. We'll also touch on DNA vaccines, a pretty remarkable application of these delivery methods. Get ready, because we're extracting the most important nuggets of knowledge and some truly surprising facts from the sources you've shared with us. 1. The foundations of gene therapy delivery viral vectors. Let's begin our journey with viral vectors, specifically lentiviruses. For many, this might be a surprising fact. Some of the most common ones used in gene therapy are actually derived from retroviruses like HIV-1. The thought of repurposing such a potent pathogen for healing immediately sparks curiosity. How do you take something so devastating and turn it into a therapeutic tool?
It's an incredible feat of bioengineering, really. Lentiviruses are enveloped RNA viruses. You find them in most vertebrates. Their natural infection cycle is, well, highly efficient. The virus binds to a host cell, its RNA genome gets inside, converts to DNA, and then integrates itself directly into the host cell's own genetic material. It becomes what we call a provirus. This provirus then basically hijacks the cell's machinery to make more viral particles. This natural ability, this efficiency in delivering and integrating genetic material is precisely what makes them attractive once they're disarmed, of course.
Okay, let's unpack this. It sounds like a sophisticated hijacking of our own cells. So if it's designed to be so effective at infecting and integrating, how do scientists actually disarm a virus like HIV-1? How do they make it a safe and effective gene therapy tool instead of, a dangerous pathogen?
The key is meticulous modification. We essentially strip away all the viral genes that make HIV-1 dangerous, the ones responsible for causing disease. We leave behind only the bare essentials needed for delivery and integration. Think of it like taking a high-performance sports car, removing the engine and all the dangerous components, and then installing a new, harmless engine that simply carries precious cargo. Crucially, any viral proteins still needed for the delivery process are provided in trans. That means they come from separate, harmless sources. This prevents the recreation of a functional, dangerous virus. So this ensures the vector can deliver the therapeutic gene, but it absolutely cannot replicate or cause illness.
That's brilliant. So you're taking a complex, dangerous machine and stripping it down to just its necessary delivery components. Now, HIV-1 naturally infects specific immune cells, right? CD4 cells. So if it's that specific, how do they broaden its reach for gene therapy? How do you target different types of cells, maybe liver cells or brain cells?
That's another ingenious trick. We pseudotype the virus. We basically replace the original HIV envelope protein, the part that dictates which cells the virus can infect, with a protein from a different virus. Most commonly, it's vesicular somatitis glycoprotein, or VSVG. VSVG has what we call broad tropism. It means it acts like a universal key, allowing the modified HIV vector to enter a wide variety of cell types. So the virus keeps its incredibly efficient delivery mechanism, but its entry key is swapped out for one that opens many more doors. This lets us target therapeutic genes to almost any cell in the body, which is pretty powerful.
That's an incredible transformation, turning a dangerous virus into such a precise delivery tool. Now let's transition to another fascinating viral vector, simian virus 40, or SV40. Its discovery is quite unusual, isn't it? Found as a contaminant in monkey kidney cell cultures, which were, ironically, being used for poliovirus vaccine production. It sounds like a problem that serendipitously became a valuable tool. What's the core story here?
Indeed, is a classic example of, accidental discovery leading to scientific breakthroughs. SV40 is a very small DNA virus. It's notable for its compact circular genome, which is actually pretty easily modifiable. In its natural replication cycle, it relies heavily on host cell machinery and produces these early proteins crucial for its life cycle. However, one of these, the large T antigen, can bind to human tumor suppressor genes like p53, and that binding potentially could lead to cancer. That was the big concern.
Okay, right. So if the wild-type SV40 has this alarming characteristic, this cancer link, how on earth do we turn a contaminant into a safe gene therapy vehicle? Is this a deal breaker, or is there a way around this major safety concern?
Oh, it's absolutely not a deal breaker, but it requires a crucial modification, a really key one. All SV40 recombinant viruses used in gene therapy are engineered to completely lack the large T antigen. Completely. This removes that primary safety concern right off the bat. Instead, we harness its efficient genome delivery and expression properties. Researchers replace those early protein genes, the problematic ones, with the therapeutic gene of interest. This ensures the virus acts only as a shuttle, delivering its cargo without any of its dangerous parts.
Okay, that makes sense. So what does all this technical detail boil down to? What are the practical features that make SV40 an attractive vector for gene therapy, despite its, slightly worrying origins?
Yeah, if we connect this to the bigger picture, SV40-based vectors offer some really compelling advantages. They can transduce both resting and dividing cells equally well. That's vital, as many target cells in the body aren't actively dividing all the time. They boast high transduction efficiency, meaning they're very good at actually getting the gene into cells. And remarkably, they offer long-lasting, maybe even lifelong transgene expression in some cases. What's also quite unique is that, to date, no significant neutralizing antibodies against the recombinant SV40 have been reported. This suggests you could potentially do multiple injections without the immune system shutting it down. That's a common problem with other viral vectors. Plus, they are quite stable and can be produced in high quantities, which is important practically.
But even with these impressive advantages, there must still be safety concerns, right? I mean, we're talking about viruses. What are the major risks researchers are constantly working to mitigate when using SV40 in gene therapy?
Yes, this raises an important question about safety, absolutely. Beyond the large T antigen issue, which, as I said, is engineered out, the main concerns are twofold. One is the potential for contamination with wild-type SV40 during production. That needs careful control. The other is the broader risk of insertional mutagenesis. Now, while this risk might be less pronounced than with some retroviral vectors, any foreign DNA integrating randomly into our genome carries a theoretical risk. It could disrupt a beneficial gene or, much less commonly, activate a proto-oncogene. So scientists mitigate these risks by using specialized production cell lines and very carefully monitoring for any unwanted viral recombination or problematic integration patterns. 2. The alternative paths, non-viral vectors.
Okay, so given those concerns with viral vectors like the immune response, the high cost, and sometimes limited DNA carrying capacity, it's pretty clear why scientists would seek out alternative paths. And that brings us to non-viral vectors, which sound like a crucial alternative.
Indeed. The promise of non-viral vectors is significant. They are generally much safer in terms of reduced pathogenicity and a lower capacity for that unintended integration, that insertional mutagenesis. They also tend to come with lower production costs and are often easier to manufacture. However, here's their main limitation currently. They often struggle to achieve really efficient gene delivery and, importantly, sustain gene expression without disturbing the host's own gene expression and signaling pathways. It's this balance between safety and efficiency that remains their primary challenge.
Right. Let's explore some of the more, let's say, conventional ways scientists have tried to get these therapeutic genes into cells using non-viral methods. Starting with the physical approaches. It sounds almost like brute force, but maybe with molecular precision.
You got to think of it that way, yeah. Traditional non-viral methods fall into two broad categories: physical methods and chemical methods. Under physical methods, we have things like hydrodynamic pressure, which is essentially injecting DNA really rapidly into a blood vessel to physically force it into surrounding tissues. It's limited, though, to areas that can tolerate temporary pressure spikes. Electroporation. This uses controlled electric shocks to temporarily create tiny pores in cell membranes, allowing DNA to slip inside. It's more common in labs, but new devices are allowing in vivo use in specific tissues like muscle or tumors. Then there's the gene gun, or particle bombardment, basically firing DNA-coated microscopic metal particles directly into cells. Pretty useful for superficial tissues like skin. And microinjection, directly injecting DNA into individual cells. It's highly precise, but very laborious and mostly used outside the body, like for modifying cells ex vivo. Then we have the chemical methods, primarily using cationic, so positively charged, molecules. Liposuction. This involves mixing DNA with cationic lipids to form tiny DNA-encapsulating spheres called liposomes. These bind to the negatively charged cell membrane and get absorbed. A challenge here is their rapid clearance from the bloodstream and potential toxicity in vivo. Cationic polymers. Molecules like polyethylenamine, or PEI, it's highly positive, so it can compact DNA into small complexes. PEI is particularly clever because it acts as a proton sponge. It causes endosomes inside the cell to swell and burst, releasing the DNA into the cytoplasm. We'll probably talk more about that. And finally, receptor-mediated delivery. This is a more targeted approach. It involves attaching a cell-specific tag or ligand to the DNA complex, guiding it specifically to cells with a corresponding receptor for precise uptake.
Okay, so these methods sound promising in theory, definitely offering alternatives to viruses. But what's holding them back in practice? Especially for real-world application in a living organism, beyond just a petri dish, it feels like the body must have so many defenses.
This really raises an important question. The common inefficiency of many of these non-viral vectors in vivo stems from several key biological hurdles. Firstly, once injected, the DNA complex can interact undesirably with blood proteins, or it might get trapped in non-target tissues, never actually reaching its intended destination. That's a big one. Secondly, even if it gets into a cell, it often gets trapped within cellular vesicles called endosomes and just fails to escape into the cytoplasm where it needs to be. Thirdly, once it is in the cytoplasm, it's vulnerable to degradation by the cell's own enzymes. And finally, getting the DNA through the double-layered nuclear membrane into the nucleus itself where it needs to be to function is another major barrier.
So if those traditional non-viral methods face all these hurdles, how are scientists improving them? How do you overcome these biological barriers and make them more effective? It sounds like they need to become smarter.
Exactly. Smarter is a good way to put it. To overcome these barriers, scientists are developing what are called novel modular vectors. These are engineered to be much more sophisticated. They often have reduced stickiness to non-target proteins, allowing them to travel better and reach their destination. They include specific zip codes, ligands for that receptor-mediated uptake by target cells we mentioned. They have elements built in to compact the DNA very efficiently, and crucially, sequences that help disrupt endosomes to release the DNA. And often, they contain nuclear import signals to actually guide the DNA into the nucleus. The real beauty of these modular vectors is their adaptability. You can sort of mix and match components. They can be customized to mimic the ability of viruses to overcome specific cellular barriers, but, without the viral baggage.
That's a clever way to make non-viral methods more virus-like in their efficiency, sort of borrowing the best bits. But even with successful delivery, we've talked about the problem of insertional mutagenesis with viral vectors like SV40. How can we ensure our therapeutic genes land in a safe place in the genome using these non-viral methods, or at least avoid unintended consequences?
This is an absolutely critical challenge. The ultimate goal is to direct therapeutic genes into what we call safe genomic locations. These are spots in the genome that won't disrupt essential cellular functions or, importantly, promote tumor growth. One really promising approach uses site-specific recombinases, or SSRs. These are enzymes, often borrowed from bacteria or their viruses, phages. They recognize unique, predefined sequences in DNA almost like a molecular GPS system. They can then cut and paste a gene precisely into that specific location. So by combining SSRs with non-viral vectors, scientists are aiming for much safer and more predictable integration. This helps ensure stable, long-term expression of the therapeutic gene without disrupting other things.
So these are like molecular GPS systems for DNA. That's a great analogy. But how do they find the right spot in our incredibly complex human genome? Do we actually have these GPS coordinates naturally in our DNA that they can recognize?
We do, but they're not perfect matches, unfortunately. Our human genome contains what are called pseudosites. These are sequences that resemble the specific recombination sites that these bacterial SSRs are programmed to recognize, but they aren't identical. So while an SSR like AC31 that's from a bacterial phage can integrate a gene at a human pseudosite, it's generally not as efficient as it would be at its native site in bacteria. That said, AC31 is currently considered the most efficient non-mammalian SSR identified for potential human gene therapy. It's capable of integrating quite large genes, like the ones needed for muscular dystrophy or certain severe skin disorders. But improving its targeting and efficiency is still a really active area of research. Now, an attractive alternative to these prokaryotic SSRs is using something from a mammalian virus, the adeno-associated virus, or AAV. AAVs are naturally non-pathogenic mammalian viruses that use a viral protein called Rep to target therapeutic genes into a specific safe harbor site on human chromosome 19. This spot is called AAVS1. This natural, precise targeting ability is very appealing for safety. The challenge, though, is that continuously producing that Rep protein inside the cell can actually be toxic. So researchers had developed ways to provide rep only transiently or perhaps from a separate source in a very controlled manner. The idea is to ensure it does its job of guiding the integration accurately, but without causing long-term cellular stress.
Okay, so we've explored methods that aim for precise integration into the genome. But what if we could achieve gene therapy without permanent integration at all? Let's introduce the concept of using DNA molecules that can replicate autonomously as extra chromosomal units. This sounds like it could avoid the very problem of insertional mutagenesis.
This is a truly elegant potential solution to bypass the risks associated with integration altogether. Naturally, some DNA viruses like Epstein-Barr virus, EBV, exist and replicate as separate, self-replicating DNA molecules, we call these episomes, inside mammalian cells. They even manage to pass copies to daughter cells during cell division. Scientists have cleverly leveraged this by creating episomal plasmid vectors. These contain specific elements borrowed from these viruses that allow the plasmid to replicate independently outside the host chromosomes. This offers the potential benefit of sustained therapeutic gene expression, but without those integration risks we've been talking about.
That sounds like a really clever workaround to the integration problem, offering the benefits of sustained expression without the risks. But what's the catch? Is there a catch? Are there any safety issues with these episomes, especially if they rely on viral components to replicate?
You're right, there often is a catch in biology. The viral proteins required for some of these episomes to replicate effectively can sometimes interfere with crucial host cell processes. This includes potentially interfering with vital tumor suppressor pathways like P53, which we mentioned with SV40. So this interference presents its own set of safety concerns, limiting their direct clinical application in some cases. However, scientists have developed smarter episomal vectors to try and get around this. A good example is the PEPI plasmid. This plasmid includes not just a viral origin, but also a sequence called a scaffold matrix attachment region, or SAMR, which is actually taken from the human genome. This SMR sequence interacts with the cell's own internal scaffolding structure, the nuclear matrix. This interaction helps the PEPI plasmid replicate and be passed to daughter cells more reliably, without relying solely on those potentially problematic viral proteins. It's a fascinating example of biomimicry learning from nature's own organizational principles to build better tools.
That sounds like a significant step forward, using our own cellular machinery. And if we're talking about ultimate cargo holders, what about human artificial chromosomes, or HACs? You described them earlier as replicas of normal chromosomes. How can such large, complex structures even be used as vectors, and what kind of cargo can they hold?
HACs are indeed the ultimate cargo holders, theoretically. They are essentially miniature, functional versions of our own chromosomes, designed to replicate and be retained within host cells, just like a natural chromosome. They possess the three critical components: a centromere for proper segregation during cell division, telomeres at the ends for protection, and an origin of replication for self-copying. Because they mimic natural chromosomes so closely, HACs offer exceptionally high cloning capacity. Think of them as supersized delivery trucks. This makes them excellent for delivering very large genes, or even multiple genes simultaneously. For instance, they've been explored for delivering the very large genes needed to potentially treat cystic fibrosis or Lesch-Nyhan syndrome. The challenge, of course, as you might guess, is getting such a massive structure into cells efficiently. This often requires direct physical methods like microinjection, which isn't always practical in vivo.
Okay, so even if we successfully get the therapeutic gene into the cell, whether integrated or as an episome, the body has incredibly clever ways to silence foreign invaders, right? Whether they're viral or non-viral. What mechanisms does our eukaryotic system use to prevent foreign gene expression, and how do we counter them to ensure our therapeutic genes stay active long-term?
This is an absolutely fundamental challenge for achieving stable, long-term gene therapy. Our cells naturally defend themselves by silencing foreign DNA. Our primary mechanism is DNA methylation. The cell adds chemical tags, methyl groups, to the foreign DNA, which essentially clamps down on the gene, preventing its expression. It marks it as off. To counter this silencing, researchers employ several clever strategies. They design smarter promoters, the on switches, for genes that can drive strong, long-term expression, even in the face of silencing mechanisms. They include those SMR sequences we just discussed, which can help create a protected environment around the gene, keeping it active regardless of where it lands in the genome. And a particularly clever approach for non-viral therapy involves creating minicircle DNA. This means stripping away the bacterial backbone part of the plasmid, which can trigger immune responses and silencing, leaving only the therapeutic gene and its essential control elements. This seemingly simple change has actually led to vastly improved and more sustained gene expression in many studies. 3. Special application DNA vaccines.
All right, so we've covered gene correction in depth. Let's pivot now to a special application of gene delivery: DNA vaccines. The history of vaccines itself is a revolution, fundamentally altering how we confer immunity. We've seen various types of vaccines over the centuries, but DNA vaccines represent a completely new paradigm, don't they?
Yes, nucleic acid vaccines, both RNA and DNA, are a truly modern approach. They work very differently from traditional vaccines. For DNA vaccines, the core idea is injecting manipulated DNA material directly into an organism. This DNA encodes a specific antigen, usually a protein from a pathogen, like a virus or bacterium. The host cells then take up this DNA, read the instructions, and produce the pathogen's protein themselves. The body's immune system then recognizes this protein as foreign and generates a specific immune response against it. It's basically immunity triggered from within your own cells manufacturing the target.
They sound incredibly promising, especially because, as the sources mention, they're very cost-effective and stable. But if they're so great, why aren't they widely used for humans yet? We see some approved for animals, but not commonly for us. What are the key advantages that still make them such an active area of research despite that?
Well, their advantages are quite compelling, which is why research continues so actively. They are generally easy and cheap to construct, compared to traditional vaccines like attenuated viruses or purified proteins. That's a big plus for manufacturing and cost. They're also remarkably stable at room temperature. This massively simplifies storage and distribution, which is particularly important for global health initiatives, avoiding complex cold chains. And crucially, they tend to favor cellular immunity. This involves T cells that kill infected cells, which is vital for fighting intracellular pathogens like viruses, where antibodies alone might not be enough. While they haven't yet consistently delivered the expected potency in human trials compared to some other vaccine types, their potential for cost-effectiveness, stability, and directing specific immune responses keeps the field incredibly active.
That distinction between antibody immunity and cellular immunity is really key, isn't it? What's the core mechanism that allows DNA vaccines to produce this crucial cellular T cell response, the CTL response, especially compared to other vaccine types?
This is a fundamental concept in immunology, and it's really why DNA vaccines were so exciting initially. Most traditional vaccines, like inactivated proteins or toxoids, primarily trigger an antibody response. B cells make antibodies. But to effectively fight many viruses, you need those cytotoxic T lymphocytes, or CTLs, which specifically recognize and kill infected cells before the virus can replicate further. For CTLs to be activated, the antigen, the foreign protein piece, has to be presented on the surface of the cell via special molecules called MHC class I molecules. This MHC class I pathway is naturally engaged when a cell gets infected by a live virus and starts producing viral proteins internally. The genius of DNA vaccines is that by having the host cell produce the viral antigen internally from the delivered DNA instructions, it essentially mimics a natural viral infection at the cellular level. The synthesized protein enters the cell's natural processing pathway and gets presented on MHC class I molecules, leading to that crucial CTL response. It's a way to potentially get the strong cellular immunity of a live virus vaccine, but without the risk of using an actual, potentially harmful live virus.
That makes sense. It's mimicking the process of infection without the danger. So what are the inherent features of DNA vaccines that allow them to mimic this viral efficiency? And what are the ongoing debates about their use that are still being worked through by researchers?
Well, the ability of a simple piece of circular DNA, a plasmid, to actually enter a mammalian cell and instruct it to produce a protein was initially quite a surprise, frankly, given how complex natural viral delivery mechanisms are. But the convenience of using DNA plasmids as gene delivery vehicles is high. They primarily just need a suitable promoter, an on switch, that's active in mammalian cells. However, despite these advantages, the safety of DNA vaccines remains a topic of ongoing discussion and research, especially regarding that theoretical risk we touched on earlier. The possibility of the plasmid DNA integrating into the host genome, even if it's rare.
Okay, and what have the real-world human trials told us about DNA vaccines' efficacy so far? Have they lived up to that initial preclinical promise in people?
The journey through human trials has been, well, complex. It hasn't been a straightforward success story yet. While many trials have definitely shown that DNA vaccines can induce detectable immune responses in humans, both antibody and T cell responses, these responses haven't always been as robust or as protective as hoped for, especially compared to the strong results often seen in preclinical animal studies. For example, some early HIV vaccine trials generated measurable T-cell responses, but unfortunately they didn't seem to affect the patient's viral counts. No clinical benefit. This led to strategies like heterologous prime boost, That's where you might use a DNA vaccine to prime the immune system initially, and then follow up with a different type of vaccine, like an adenoviral vector, to boost the response much higher. More recently, a lot of the DNA vaccine research focus seems to have shifted towards targeting tumor antigens in cancer immunotherapy, possibly driven by some promising results and perhaps funding opportunities in that area.
Right. So, beyond the scientific mechanism itself, how are these vaccines actually administered? What are the practical delivery techniques? And how do they impact effectiveness if just injecting the naked DNA isn't always optimal?
Yeah, due to those somewhat discouraging initial clinical trial results, there's been a really strong focus on enhancing the delivery methods. DNA vaccines are typically given either intramuscularly, IM, into the muscle, or intradermally, ID, into the skin. The IM route into muscle tends to get higher levels of antigen expression because muscle cells are good factories, but muscle tissue is relatively less immunogenic because it lacks many of those crucial antigen-presenting immune cells, APCs. The ID route into the skin usually results in less antigen expression overall, but it's often much more immunogenic. Why? Because the skin is absolutely packed with those APCs, like dendritic cells and Langerhans cells, which are essential for kickstarting a strong immune response. It's like delivering the message right to the immune system's front door. To boost efficiency further, devices like the gene gun, which literally shoots DNA-coated gold particles into the skin at electroporation using brief electric pulses to temporarily open cell membranes, are often employed. These are sometimes called second-generation DNA vaccines. Researchers are also exploring packaging the naked plasmid DNA, pDNA, into nanoparticles made of lipids or polymers. This can help increase the pDNA stability in the body and improve its uptake by cells.
Okay, so if the current results aren't always as robust as hoped, even with better delivery, how are scientists trying to boost the effectiveness of the DNA vaccines themselves? What are the enhancement strategies at play to make them more potent immunologically?
That's a great question. Several clever strategies are being employed at the molecular level. One approach is enhancing the immunogenicity of the encoded antigen itself, making it more visible or stimulating to the immune system. This can be done by adding a genetic adjuvant. Here, the plasmid carries not just the gene for the antigen, but also a gene for a protein known to boost immune responses. They get co-expressed in the same cell, providing extra kick. Another way is to actually alter the gene encoding the antigen. Codon optimization is a big one. This involves editing the gene sequence, changing some DNA letters without changing the final protein so that it's more efficiently and translated into protein by human cell machinery. This can significantly boost the amount of antigen produced. You can also add signal sequences to the gene. These act like postal codes, directing these synthesized antigen protein to specific compartments within the cell, like the endoplasmic reticulum, which can improve its processing and presentation on those MHC molecules we talked about, making it more likely to trigger T cells. Sometimes, the antigen gene is even genetically fused to the gene for an entire carrier protein, known to strongly engage the immune system, effectively making the target antigen much more noticeable.
Fascinating strategies. Now let's circle back to the critical question. How safe are DNA vaccines really? What are the potential risks that researchers are monitoring, especially regarding that potential for integration into our own genome?
Generally speaking, DNA vaccines are considered to have a pretty good safety profile. In trials, the main side effect reported is usually just limited local reactivity at the injection site, a bit of redness or soreness. The main theoretical concern, the one that always comes up, is that the plasmid DNA genome might potentially integrate into the host cell's DNA. If this happened randomly, it could theoretically disrupt an important gene or, in the worst-case scenario, activate an oncogene, potentially leading to cancer down the line. Now, to assess this risk realistically, scientists compare the measured rate of plasmid integration to the body's natural background rate of spontaneous mutations in our own genes, which is known to be very low, but not zero. While extensive human studies looking specifically for integration are difficult to perform routinely, you need biopsies, animal studies have consistently shown that the integration rates for DNA vaccines are always several folds lower than the spontaneous mutation rate of our own genes. This suggests the actual risk is likely very low, but it's definitely an area that requires ongoing monitoring and careful vector design.
Okay, that puts the risk in perspective. Beyond direct human vaccination, what are some of the other, perhaps you could call them secondary roles that DNA vaccines are playing in biological research and medicine? They sound like versatile tools.
Oh, they're incredibly versatile research tools, absolutely. They're used quite widely in labs. For instance, they're used to immunize animals to generate specific monoclonal and polyclonal antibodies for research or diagnostic purposes. They're used in the process of creating knockout mouse models, where a gene is inactivated to study its function. And researchers can develop DNA vaccine libraries, collections of plasmids encoding many different potential antigens from a pathogen to screen and identify which ones actually confer protective immunity, even without knowing much about the proteins beforehand. And while we focused heavily on DNA, it's definitely worth noting again that RNA vaccines, particularly mRNA vaccines and self-amplifying RNA, are gaining huge prominence. They offer ways to potentially overcome some of the limitations seen with plasmid DNA, like the integration concern and sometimes lower potency, as we've witnessed dramatically in recent global vaccination efforts. 4. The tiny carriers lipoplexes and polyplexes.
Right, we've covered viral delivery and DNA vaccines extensively. Now let's transition to another really crucial category of non-viral gene delivery, lipoplexes and polyplexes. We've already established the formidable challenge of delivering nucleic acids effectively, getting them to specific target cells and tissues. These tiny carriers offer yet another distinct approach.
That's right. These non-viral methods, lipoplexes using lipids, essentially fatty molecules, and polyplexes using polymers, which are long-chain molecules, are appealing alternatives to viral vectors. They aim to avoid those issues like immunogenicity and limited DNA carrying capacity that can plague viruses. They operate fundamentally on electrostatic interactions. They are designed to be positively charged, which allows them to bind to the negatively charged surface of our cell membranes and then hopefully get absorbed or taken up. A key challenge, however, as with many non-viral methods, is their efficiency in vivo, especially getting the DNA payload past all the cellular defenses and successfully into the nucleus where it needs to function.
So what are the key factors that scientists really need to consider to make these delivery systems successful? What determines if the DNA actually gets where it needs to go and expresses effectively?
Yeah, for successful DNA delivery using these systems, several critical factors absolutely must be optimized. It's a complex puzzle. You need to control the carrier's nanoscale structure, how the lipids or polymers assemble with the DNA. You need to understand precisely how they interact with the cell membrane. Their exact chemical composition matters hugely. Their pathway for getting into the cell, the internalization route, is critical. And crucially, how efficiently they release the DNA cargo once they are inside the cell. Each one of these steps is a potential bottleneck that can limit the overall success.
Okay, so once scientists understand their structure and properties, what's the actual journey of these nano-sized complexes once they encounter a cell? How do they manage to get the DNA payload inside and then release it in the right place? It sounds like a multi-step obstacle course.
It really is an intricate journey. First, the positively charged lipoplex or polyplex binds to the negatively charged cell surface. That initial electrostatic interaction is key. Then the cell internalizes the complex, usually by engulfing it in a membrane-bound sac or vesicle called an endosome. This process is called endocytosis. Now, this is where a major hurdle occurs. The complex must escape from the endosome before the cell degrades it. It needs to get into the main body of the cell, the cytoplasm. For lipoplexes, helper lipids like DOPE are often included because they can help destabilize the endosome membrane, kind of punching holes to let the DNA out. For polyplexes, especially those containing PEI, they use that clever proton sponge mechanism we mentioned. As the endosome naturally tries to acidify itself, the polymer soaks up the protons. This disrupts the salt balance, causing water to rush in, making the endosome swell and eventually burst, releasing the complex. Okay, so once it's free in the cytoplasm, the DNA needs to dissociate from its carrier, the lipid or polymer. It has to break free to be useful. And finally, the naked DNA must somehow import into the nucleus. This can happen passively during cell division, when the nuclear membrane temporarily breaks down, or potentially through active transport mechanisms via nuclear pores. This nuclear uptake step has historically been a really significant barrier for non-viral gene delivery.
That's a fascinating journey, full of challenges. Let's dive deeper now into the first type, lipoplexes. What exactly are they made of, and how are they structured to effectively deliver their genetic cargo?
Right, lipoplexes. They're formed by mixing DNA, which is negatively charged, with cationic lipids, which are positively charged, often along with neutral helper lipids like DOPE or cholesterol. The cationic lipids are amphiphilic molecules, meaning they have a water-loving positive head group that binds to the DNA and a water-heating fatty tail that helps form the structure of the liposome, like a tiny bubble. Helper lipids play crucial roles. DOPE, for example, exists in a structure at neutral pH that helps destabilize membranes when it encounters the acidic endosome, facilitating DNA release. Cholesterol, on the other hand, often gets added because it tends to make the liposomes more stable in the bloodstream, protecting the DNA from degradation before it reaches the target cell. The specific shape and charge of these lipid molecules, the length of their fatty tails, even the type of chemical linker connecting the head and tail, all these details are critical and are tweaked by chemists to optimize delivery. How they self-assemble with DNA is also key. For optimal function, they often need to form specific structural arrangements when they interact with cell membranes, like a hexagonal phase, which seems to be important for getting the DNA across the membrane and into the cytoplasm.
Okay, that gives us a good picture of lipoplexes. Now let's turn our attention to polyplexes, the polymer-based carriers. How do they fundamentally differ from lipoplexes, and what unique characteristics do they bring to the table for gene delivery?
Polyplexes use cationic polymers long-chain molecules with repeating positive charges, like polyethylenamine, PEI, or polyalazine, PLL, instead of lipids. The key functional difference is that these polymers typically lack the large hydrophobic domain that lipids have. This means they can't really fuse with membranes in the same way lipoplexes sometimes do. Their strength lies more in their ability to condense DNA very tightly, packaging it into extremely small, stable particles. This small size is really important for getting into cells and potentially navigating the intracellular environment. Second-generation polymers like PEI, as we've discussed, are particularly effective because of that proton sponge ability, which provides a powerful mechanism for escaping the endosome. Another big advantage of polymers is that scientists can often modify them more easily than lipids. You can readily attach other functional groups, things like peg chains, to provide stealth properties and improve stability in the bloodstream, or specific targeting ligands to guide the polyplex to particular cell types. This makes them highly customizable delivery platforms.
Right, the customization sounds powerful. So, to bring this deep dive into lipoplexes and polyplexes to a close, what's the big picture here? What have we learned about these innovative nanocarriers, and what are the main remaining challenges?
To summarize, I think it's clear that both lipoplexes and polyplexes can be rationally designed and optimized to function as efficient non-viral vectors for gene therapy. The ultimate goal remains the development of an ideal gene delivery vector, one that's perfectly tailored for in vivo use, highly stable in the body, easily transported across biological membranes, and highly efficient at delivering its payload. We've learned that specific structural features matter, like lipoplexes transforming into that hexagonal H11 phase, seem to show higher transfection activity. A practical challenge is that many effective lipoplex formulations, especially those containing DOPE, tend to aggregate in blood, limiting their use for systemic applications, although they work well in cell culture. So serum-resistant formulations are a key area of development. For polyplexes, achieving complete condensation of the DNA offers key advantages like protection from degradation and enhanced cellular uptake. But perhaps the main challenge that still remains for both systems is ensuring the efficient dissociation of the DNA from its carrier once it's inside the cell, particularly once it reaches the nucleus. The DNA has to break free to become transcriptionally active. So there's a continued strong need for rational design and clever chemistry to create vectors with ever-improving transfection ability and release characteristics. 5. The bare essentials naked DNA delivery.
Okay, from complex nanocarriers like lipoplexes and polyplexes, let's simplify things for a moment. What about just naked DNA? This is described in the sources as the simplest non-viral gene delivery system, highlighting its directness. But does that simplicity actually translate to efficacy in practice?
Yeah, naked DNA delivery is exactly what it sounds like. Basically, direct injection of the vector DNA, usually a plasmid, directly into a cell or tissue. It's the most straightforward approach. While it does lead to some level of gene expression, it's important to be clear that the levels achieved are generally much, much lower than what you typically get with viral vectors or even optimized liposomal or polyplex vectors. Its major limitation is its vulnerability in the body. It gets rapidly degraded by enzymes called nucleases that are present in our bloodstream and tissues. This makes it pretty much unsuitable for systemic administration. You can't just inject it into the blood and expect it to reach a target organ intact. So its application is mostly limited to easily accessible tissues, like muscle cells or the skin. And even there, the efficiency of simple DNA injection into muscle, despite a lot of research, hasn't really proven efficient enough for widespread clinical level use on its own.
Right. So, despite these significant limitations, naked DNA must have found its niche where it is proving effective, or at least useful. Where's this seemingly simple approach making a difference?
Absolutely. Despite its limitations, naked DNA has definitely found success in specific contexts, particularly where the target tissue is directly accessible, bypassing that degradation issue in the bloodstream. For example, it's being used quite a bit in cancer therapy research. DNA encoding tumor antigens can be injected directly into a tumor, or perhaps into muscle, to express those antigens and hopefully stimulate an immune response essentially functioning as a type of cancer vaccine. It's also being applied experimentally to treat certain genetic diseases in directly available tissues, like treating skin disorders by injecting DNA into the skin. Importantly, advances in delivery techniques are helping to boost its practicality. Methods like electroporation, which we discussed earlier, used alongside the injection, can significantly enhance DNA uptake into muscle and skin. And surprisingly, under certain conditions, intravenous injection can result in relatively high gene transfers specifically to liver cells in animal models. So it's now being explored in many clinical trials, for example, in treating peripheral arterial occlusion disease by injecting genes to promote blood vessel growth directly into leg muscles.
Okay, so delivery methods like electroporation can help. But once this naked DNA gets into a cell, what causes it to eventually be lost, or for the gene expression to diminish over time? The body seems to have a lot of ways to counteract our efforts, doesn't it?
It really does. The body has strong defenses and cleanup mechanisms. Plasmid DNA delivered this way can be lost for several reasons. Sometimes the injection process itself can cause local cell damage or inflammation, and transgene expression might vanish simply due to cell death in that area. A major factor is that the plasmid DNA usually doesn't integrate into the host cell's genome. So, as the cells divide, the plasmid gets diluted out among the daughter cells and eventually lost. Also, the promoter sequences driving gene expression on the plasmid can sometimes become inactive over time, silenced by the cell's own regulatory mechanisms. But a very important way of losing plasmid DNA, especially relevant for DNA vaccines, is via the immune response. Our immune system is very good at recognizing bacterial DNA, which is the source of most plasmids used in research as foreign. This is largely because bacterial DNA contains sequences called unmethylated CpG motifs, which are much rarer in our own DNA. These CpG sequences trigger specific immune sensors like toll-like receptor 9, TLR9, leading to inflammation and clearance of the foreign DNA, effectively shutting down gene expression. So a key strategy in designing better plasmids is to minimize these CpG sequences as much as possible to try and evade this immune detection and prolong gene expression.
That immune recognition point is really interesting. Now, a quick but important side note on a related concept mentioned in the sources: small interfering RNA or siRNA. This offers a different kind of gene modulation, right? Not by adding genes, but by silencing them.
Indeed. RNA interference, or RNAi, is a really powerful biological process that cells use naturally, and scientists have harnessed it. It uses small interfering RNA, or siRNA. These are tiny, typically 21-25 base pair long, double-stranded RNA molecules. When introduced into a cell, siRNA can specifically find and trigger the destruction of a targeted messenger, RNA, mRNA, molecule, effectively knocking down or significantly reducing the expression of the corresponding gene. It stops the protein from being made. This is a powerful tool to specifically turn off problematic genes that might be causing disease. It's been shown experimentally to reduce protein expression in targeted tissues by up to 90%, with potential applications in lung, heart, liver diseases, and many others. The trick with siRNA, however, is similar to naked DNA delivery and stability. Its small size makes it prone to degradation and leakage, especially during systemic delivery. Ensuring it stays in the target tissue long enough to have a therapeutic effect is still a major challenge being actively worked on. 6. The jumping genes transposons.
Okay, we've explored highly engineered viruses and a whole variety of non-viral methods, from physical zapping to tiny carriers and even naked DNA. Now, let's lead into a truly exciting and, as the sources say, varied mechanism for non-viral gene delivery: transposons. These are often referred to as jumping genes. This sounds like a completely new paradigm altogether.
It absolutely is a different paradigm. Transposons are fascinating mobile genetic elements that exist naturally in almost all organisms. They operate basically by either a cut and paste or sometimes a copy and paste mechanism to move themselves around within the genome. For gene therapy, we focus on the cut and paste type. The key player is an enzyme called a transposase. This enzyme recognizes specific sequences on the transposon DNA, cuts the transposon out, carrying the therapeutic gene we've inserted into it, and then helps integrate it into a new location in the host cell's chromosomal DNA. The two systems that are most commonly used and studied for genetic modification in human cells are called Sleeping Beauty, often abbreviated SB, and Piggyback.
Jumping genes, it's such a great name. What makes them so appealing? What distinct advantages do they offer over other gene delivery methods we've discussed, especially compared to the viral vectors?
The advantages of using transposons are really quite compelling, which is why there's so much excitement around them. Firstly, they are generally much easier and significantly cheaper to produce at clinical grade compared to viral vectors. Manufacturing viruses safely and consistently is notoriously expensive and complex. Transposon plasmids are simpler. They also typically have fewer storage problems. Plasmids are more stable than many viral preparations. And crucially, they tend to exhibit less innate immunogenicity. They don't usually trigger the same kind of strong, immediate immune response that viral vectors can, which improves their safety profile. What's more, they offer the potential to easily deliver large or even multiple genes simultaneously. This overcomes a major limitation of many viral vectors, which have a fairly restricted cargo capacity. They've already shown really good results in genetically modifying various clinically relevant cell types, including human T lymphocytes for cancer therapy, and various types of stem cells, including induced pluripotent stem cells, iPSCs. This opens doors for treating complex genetic disorders and developing advanced cell therapies. Just for a little bit of historical context, these jumping genes were actually first described way back in the 1940s by Barbara McClintock, studying mosaic patterns in corn kernels, a discovery that eventually won her the Nobel Prize. And it's quite remarkable, really, that estimates suggest around 47%, almost half, of our own human genome is derived from ancient transposon activity over evolutionary time. As I mentioned, they operate via two main mechanisms. Class I are retrotransposons. They copy and paste using an RNA intermediate. But for gene therapy, we mostly use Class II, the DNA transposons. These cut and paste. The transposase enzyme recognizes specific sequences called inverted terminal repeats, IRs, that flank the transposon DNA, excises the whole segment, and then helps paste it into a new spot in the genome, often by creating double-stranded breaks at the target site.
Fascinating history. Let's dive into the specifics now of the two leading transposon systems you mentioned, Sleeping Beauty and Piggyback. What's the story behind Sleeping Beauty, and how has it been engineered or adapted for therapeutic use?
Sleeping Beauty, or SB, has a really cool origin story. Its functional gene sequence was actually computationally reconstructed, essentially resurrected, from inactive, fragmented transposon sequences found scattered in the genomes of salmonid fish, hence the evocative Sleeping Beauty name, as it was effectively awoken by scientists. It belongs to a large superfamily called TC1 Mariner transposons. For gene therapy, the system consists of two components delivered to the cell. One, the therapeutic gene of interest, cloned inside the transposon element, which is flanked by those IR sequences. And two, a separate source providing the Sleeping Beauty transposase enzyme, which makes the transposon jump from its delivery plasmid into the host cell's chromosome. SB has shown considerable promise in preclinical studies for treating a range of conditions, like the skin disorder epidermolysis bullosa, sickle cell anemia, and even certain cancers like glioblastoma. Researchers are constantly working to refine the system, engineering more active versions of the transposase, and optimizing the delivery methods to boost its efficiency and ensure its safety by better understanding and potentially controlling where it integrates.
A resurrected gene from fish. That's amazing. Now let's turn to Piggyback. What's its origin, and what's a truly significant feature that sets it apart, perhaps even from Sleeping Beauty?
Piggyback is another Class II transposon, originally isolated from the cabbage flooper moth, Trichoplusionia. Its truly significant and distinguishing feature is its remarkable ability to excise itself from the donor site without leaving any footprints or mutations behind. When the piggyback transposase cuts the transposon out, it does so in such a way that the original DNA sequence at the donor site can be perfectly regenerated, often through simple ligation of the resulting DNA ends. No extra bases added or deleted. This no-footprint characteristic is a huge advantage for safety, especially for applications like cellular reprogramming or transient gene expression, where you might want to introduce genes temporarily, use them for a specific purpose, and then remove them cleanly without permanently altering the original cell's DNA sequence.
Wow, that no-footprint feature sounds almost revolutionary for safety and precision. So, comparing these two leading systems directly, Sleeping Beauty and Piggyback, what are the key differences in their capabilities and characteristics that researchers need to weigh when choosing one over the other for a specific application?
That's a crucial comparison. They have distinct strengths and weaknesses. Cargo capacity. This is a big one. Sleeping Beauty's efficiency tends to drop off significantly with cargo sizes much larger than about 10 kilobases, kb. Piggyback, on the other hand, has demonstrated the ability to mobilize much larger genes, potentially exceeding 100 kilobases in some studies. That's a massive difference if you need to deliver very large genes or multiple genes together. Footprint upon excision. As we just discussed, SB typically leaves a small 3-base pair mutation footprint at the insertion site when it excises. Piggyback, remarkably, leaves no mutation, offering that clean excision. Integration site preference. This is subtle but important. SB integration appears to be relatively random across the genome. Piggyback, interestingly, shows somewhat of an increased preference for integrating within active genes in near transcriptional start sites, TSS. Whether this is an advantage or disadvantage depends heavily on the specific therapeutic context and safety considerations. Modifiability for engineering. Piggyback's transposase seems more tolerant to having other protein domains fused to it without losing much activity. This makes it potentially more flexible for engineering things like site-directed integration systems. SB can be more sensitive to such modifications. Both systems also have engineered hyperactive versions available like SB100X for Sleeping Beauty and High-P base for Piggyback, which greatly boost their transposition activity, allowing researchers to use less material.
Okay, that comparison is really helpful. Let's elaborate now on those compelling advantages that truly make transposons feel like a potential game changer in gene delivery, particularly when we think about their practical application compared to, say, the more established viral vectors.
Certainly. One of the most significant practical advantages is cost in manufacturing. Producing clinical-grade viral vectors is incredibly expensive, time-consuming, and requires highly specialized facilities and rigorous testing for safety, like ensuring no replication-competent virus is present. This really limits their widespread accessibility. Transposon plasmids, being simpler DNA molecules, are much quicker and cheaper to manufacture at clinical cGMP grade. Scaling up production is also considerably easier, and existing facilities can potentially be adapted more readily. This potential to dramatically reduce both the time and the cost of producing gene delivery systems is a massive driver for the field. Another huge benefit, as we touched on, is their ability to deliver large and multiple transgenes. Viral vectors are constrained by the physical size of their capsid. They simply can't package overly large genetic payloads, usually capping at around 8-10 kilobases. While early SB systems had efficiency drop-offs beyond 10 kilobases, the piggyback system has been shown to successfully modify human cells with transposons carrying 15 kilobases, or even mobilize enormous constructs like 100 kilobase bacterial artificial chromosomes, BACs, in stem cells. This increased cargo capacity is crucial. It allows us to consider delivering not just one corrective gene, but maybe multiple genes needed for a complex pathway, or very large genes like dystrophin for muscular dystrophy, all within a single vector system. Then there's the crucial aspect of less immunogenicity. We tragically saw in early gene therapy trials, like the Jesse Gelsinger case in 1999, involving an adenoviral vector, that strong immune responses to the viral vector itself can have devastating consequences. Our immune system is highly attuned to detecting viral components. Even highly stripped-down viral vectors can sometimes trigger inflammation. Transposons, being essentially just DNA delivered non-virally, often via electroporation or similar methods, generally induce much less of this problematic innate immune response related to viral proteins or capsids. While the DNA itself can trigger some immune sensing, like via those CpG motifs, it's typically much less severe than the response to a whole virus particle. This offers a significantly potentially safer profile. And finally, there's the point about a potentially lesser tendency for oncogenic mutations due to integration patterns. Some retroviral vectors, like those derived from marine leukemia virus, MLV, showed a concerning preference for integrating near transcriptional start sites and enhancers. This unfortunately led to leukemia in some patients in the French XSEID gene therapy trial due to integrations near the promoter of the LMO2 proto-oncogene activating it. Genome-wide mapping studies of Sleeping Beauty integrations in mammals show some preference for transcriptional units, but it seems less strongly biased towards TSS than MLV, and it can vary between cell types. Piggyback integration in human cells, interestingly, appears to have no significant overall preference for specific chromosomal regions. While it does show some bias towards integrating within RefSec genes and near TSS and CpG islands, it seems less problematic overall than MLV, although this might depend on the cell type or state. Critically, both Sleeping Beauty and Piggyback are actively being engineered for site-directed gene delivery, aiming to guide integration to specific safe locations, which should dramatically improve safety further. This potential for safer integration profiles is so promising that, as mentioned, the FDA has approved the first clinical trial using transposon-modified T cells.
Okay, those are some really compelling advantages: cost, cargo size, lower immunogenicity, potentially safer integration. But as always, no technology is without its hurdles. What are the current challenges that researchers are actively working to overcome to bring these transposon systems fully into widespread clinical practice?
Yes, despite all the promise, significant challenges definitely remain. First, gene delivery efficiency into the target cells can still be a limitation. Remember, the transposon sister itself, the transposon, the DNA, and the transposon source, usually needs to be delivered into the cell using non-viral methods, often native DNA plasmids delivered by physical means like electroporation or nucleofection. While these methods can be effective, especially in some cell types grown in culture, they can also be quite harsh on the cells, leading to significant cell death, which reduces the overall efficiency of getting stably modified cells. Improving the initial delivery of the transposon components into primary human cells, especially in vivo, remains a key bottleneck. Novel methods like using cell-penetrating peptides fused to the transposes are being explored. Second, there's still the issue of the somewhat random integration profile, even if it's considered safer than some viral vectors. Uncontrolled integration, even if less biased towards dangerous spots, still carries a risk of disrupting essential genes or regulatory elements, leading to genotoxicity. Achieving reliable and highly efficient site-directed integration is paramount for ultimate safety. Third, researchers sometimes observe the silencing of the integrated transgene over time. Just because the gene got integrated doesn't guarantee it will stay active long-term. Epigenetic mechanisms can shut down the expression from the transposon, reducing the therapeutic effect. Understanding and preventing this silencing is another important area of ongoing research, especially for piggyback, where it's been less studied than for SP.
Right, those challenges are significant but seem like active areas of innovation. Despite these hurdles, the applications of transposon systems are already incredibly exciting and seem transformative in various fields of medicine. Can you give us some key examples of diseases being tackled or cellular engineering advances where they've made a significant impact already?
Absolutely. The applications are really where the excitement builds. Transposons are making real inroads. In terms of specific diseases, preclinical studies using Sleeping Beauty have shown promise for correcting models of hemophilia, both A and B, tyrosinemia type I, junctional epidermolysis bullosa, and even potentially Huntington's disease. Piggyback has been used effectively in models of alpha-1 antitrypsin deficiency. Beyond single-gene disorders, they're revolutionizing the genetic modification of human T lymphocytes for cancer immunotherapy. This is a huge area. Modifying patient T cells to express chimeric antigen receptors, CARs, that target cancer cells is a powerful new therapy. Doing this with viral vectors is expensive and complex. But transposons, particularly the Sleeping Beauty system initially, demonstrated successful modification of human T cells with CARs, generating functional CAR T cells. This work directly led to that FDA-approved clinical trial using SP-modified CAR T cells to fight B-cell lymphomas. Piggyback is also being used extensively now to stably express CARs and potentially other modifications in T cells, aiming to improve their efficacy and persistence. They're also proving transformative in the generation of induced pluripotent stem cells, or iPSCs. iPSCs hold immense promise for regenerative medicine because you can create patient-specific stem cells from easily accessible cells like skin or blood. Older methods using integrating viral vectors to deliver the reprogramming factors carried a risk of tumor formation if those factors weren't silenced or if the virus integrated poorly. Transposons offer higher gene delivery efficiency for reprogramming, and crucially, especially piggyback with its no-footprint feature, they can be excised cleanly from the cells after reprogramming is complete. This allows the generation of transgene-free iPSCs, which are considered much safer for potential therapeutic use, as you don't have residual reprogramming genes hanging around that could cause problems later. This clean removal is a major advantage over many viral methods. And more generally, transposons are being widely adopted for the genetic modification of various types of stem cells, including human embryonic stem cells and hematopoietic, blood-forming stem cells. They provide an effective mechanism for making permanent genetic modifications, or importantly, reversible modifications in the case of piggyback in these critical cell types, paving the way for developing advanced stem cell-based gene therapies for a wide range of conditions.
That's incredibly exciting, especially the iPSC application and the CAR-T therapies. Looking ahead now, what are the really cutting-edge areas of research right now? What promises to make these transposon systems even more powerful and precise in the near future?
Two areas stand out as particularly hot and promising right now. First is the continued generation and refinement of hyperactive transposon elements. Researchers have been incredibly successful in engineering supercharged versions of both the Sleeping Beauty and piggyback transposase enzymes. We have versions like SB100X and HiPBase that show activity levels potentially 100-fold or even more higher than the original natural transposes. This means you can potentially achieve the same level of gene transfer using much less transposes and transposon DNA, which can reduce costs and potentially minimize off-target effects or toxicity. The engineering involves clever strategies like borrowing beneficial mutations from related transposes or using high-throughput screening to find better variants. Second, and perhaps most critically for clinical translation, is the intense focus on engineering transposon systems for site-directed integration. As we've discussed, avoiding random integration is key for ultimate safety. The goal here is to precisely guide the transposon to insert only into specific, predetermined safe harbor locations in the genome. The main approach involves fusing the transposase enzyme to specific DNA binding domains, things like zinc finger proteins, ZFPs, or talons, or even potentially elements from CRISPR systems that recognize and bind to a unique target sequence in the human genome. This fusion protein should then preferentially insert the transposon right next to where the DNA binding domain is bound. Piggyback is often favored for this because its transposase seems more tolerant to these kinds of fusions without losing its cutting and pasting activity. There have been successful proof-of-concept studies showing biased integration near target sites using engineered piggyback transposases. The current challenge is making this targeting highly efficient and specific, minimizing off-target integrations where the transposase still inserts randomly, despite the targeting domain. But achieving reliable site-directed integration would be a massive leap forward, improving gene expression consistency and dramatically enhancing the safety profile. So overall, when you look at the low cost, the flexibility in cargo size, the reduced immunogenicity, and especially the rapid progress being made in achieving site-directed integration, transposons really do make an exceptionally promising non-viral gene delivery system for the future of gene and cell therapy.
Wow. What an incredible journey we've taken today through the really intricate world of gene therapy delivery. We started with the ingenious repurposing of viral vectors like HIV and SV40, essentially stripped down and re-engineered for healing. Amazing stuff. We then explored the really diverse non-viral strategies, everything from physical methods to those complex tiny chemical carriers like lipoplexes and polyplexes. And finally, we delved into the dynamic jumping genes, the transposons marveling at sleeping beauty and piggyback, alongside that evolving and still very promising field of DNA vaccines. It truly highlights the sheer ingenuity and the breakneck pace of advancement in this field. It's moving so fast.
It really does. And I think the core theme running through all these different approaches is that relentless pursuit of safer, more efficient, and ever more precise ways to deliver therapeutic genes into the cells where they're needed. Scientists are constantly performing this delicate balancing act, maximizing the effectiveness of gene delivery while minimizing those crucial risks, whether it's triggering an immune response, causing unintended side effects from the vector itself, or the potential danger of insertional mutagenesis. Each method we discussed today, as you heard, presents its own unique set of advantages, but also its own inherent challenges. And it's really this ongoing tension, this need to overcome limitations, that drives the continuous innovation and pushes the boundaries of what's actually possible in medicine.
Absolutely. This deep dive today really reveals a potential future where gene therapy could be precisely tailored for almost any condition, moving way beyond just fixing a single missing gene. What could be the profound societal implications, do you think, of a future where we can precisely edit and express genes in almost any cell type, almost at will? It opens up huge possibilities, doesn't it?
Indeed, the implications are potentially staggering. Consider how our fundamental understanding of human health, disease, and even aging could change when gene delivery becomes potentially as routine, as safe, and as accessible as, say, current vaccine administration is today. It raises profound questions, not just about the future of medicine, but about our very biological selves. How do we approach health and disease when we can potentially intervene at the most fundamental genetic level? We could shift from merely treating symptoms or managing chronic conditions to potentially preventing or even curing diseases at their genetic root. It's a field that could truly redefine what it means to be healthy in the 21st century and beyond.
It really could. A future shifting from treatment to cure, or even prevention at the genetic level. It's a field that truly makes you think. We encourage you, our listeners, to reflect on these incredible possibilities and continue your own deep dives into the science that is actively shaping our future. Until next time on The Deep Dive, stay curious.